Hello dear friends, welcome back. We are discussing about the muscles of the human body, especially the skeletal muscles, the anatomy of them and the locations of each muscle. In my previous lectures, I have introduced the muscles of the body, right? That what is a muscle, and we discussed about the neuromuscular junction. And now in this lecture, we are comparing the three types of muscles that we have in our body. In our body, we have some tissues which contains the elastic fibers, right? The actin and myosin filaments, right? So those are called muscles, and in our body we have three types of muscles, right? The skeletal muscles, the cardiac muscles, and the smooth muscles, right? Now let's compare all of the three muscles and the properties of all the three muscles. So the first property that we have here is the location of these muscles. That what is the location of skeletal muscles, the cardiac muscles, and the smooth muscles. We have a comparison between all of the three muscles. So first, about location. The location of skeletal muscles, that skeletal muscles in our body are attached to our skeleton, right? Cardiac muscles are present in our heart, right? And smooth muscles are present everywhere in our body. We say everywhere in our body, especially in the walls of viscera, right? The walls of organs. Then they are present in the blood vessels, the inside walls of the blood vessels, right? That the tunica media of the blood vessels are contain the smooth muscles, right? And in our eyes, the iridae muscles, dilated pupillae, constricted pupillae, all these are what? They are the smooth muscles. And beneath our skin or in our skin, like the muscle which is connected to the erector pili muscle, the erector pili muscle which is connected to the root here, right? Which makes our hairs stand erect. So those muscles are the which muscles? They are the smooth muscles. So the location, the first feature is the location. The second feature is about the cell shape. What is the shape of the skeletal muscle cells, right? Cell shape. They are long thread-like fibers, which I will show in the video. You can see right now on the screen. So they are long thread-like fibers. The cardiac muscles are short and slightly branched, which you can see on the screen. They are branched muscles. And smooth muscles are short and fusiform. They are fusiform, right? Which I am showing right now on the screen. You can see. So they are fusiform. Then what about the length of the muscles or cell? The cell of the muscle. The length of the skeletal muscle cell is from 100 micrometer to 30 centimeter, right? Longest muscle cell, 30 centimeter. The cardiac muscle cell length of them are 50 to 120 micrometer. Now you can see the difference, right? And what about the length of the smooth muscle cell? 30 to 200 micrometer, right? Then what is the cell width? Right? The difference between the width of the cell. This was the length of the cell. Now the width of the cell. The width of the cell of skeletal muscle is 10 to 500 micrometer. The width of the cardiac muscle cell is 10 to 20 micrometer, and the width of the smooth muscle cell is 5 to 10 micrometer, right? Next property is, what about the nuclei, right? In our body, we have the smooth muscles, and all the smooth muscles inside their cell contain only one nucleus. So we call them uninucleated, right? We are talking about the cell of the muscle. Then the cardiac muscle cell contain one nucleus. They are also uninucleated. And the skeletal muscle cells contain many nuclei in one cell. They are called multinucleated, right? The fifth property. The sixth property is the presence of striations. In our skeletal muscles and cardiac muscles, we have those light and dark bands, the Z lines, the M line, the H zone, the A band, the I band, right? The thin fibers, the thick fibers, all of them make light and dark bands, which you can see right now on the screen. So the light and dark bands, so those light and dark bands are called the striations, right? So all those muscles which contain all those stria, all those light and dark bands, they are called striated muscles. So we say in skeletal muscles, striations are present. In cardiac muscles, striations are present. But in smooth muscles, there are no sarcomeres present, right? In cardiac and skeletal muscle, you have sarcomeres present. But in case of smooth muscle, there are no sarcomeres present. So there will be no light and dark bands. There are no striations there, right? The next property is of the connective tissue surrounding the muscle, right? When I had opened the skeletal muscle, I told you that about the skeletal muscle there is the epimysium, right, perimysium and endomysium. All those were connective tissues surrounding the muscle, then the muscle fascicle, then the muscle fiber, right, or fibril. So these the skeletal muscle here, there are skeletal muscle. Skeletal muscles contain endomysium, perimysium and epimysium. About cardiac muscle, it only has endomysium, and about smooth muscle, it also has only endomysium, right? So this was the difference. The next difference is for T tubules. Inside the T tubules, right, when the muscle is excited, calcium runs, right? So calcium will run through the T tubule, right? So inside the smooth muscles, you have no T tubule, right, for the entry of calcium, because in these cells, the calcium will enter through the proteins, right, through the pores in the cell membrane, right? So there is no T tubule present. In skeletal muscles, we have T tubule present, but they are narrow tube, and in case of cardiac muscles, T tubules are present, but they are wider a little bit, right? So the T tubules through which calcium will enter the cell, in skeletal muscle they are present, in cardiac muscle they are present, but they are absent. Next, what about the sarcoplasmic reticulum? Inside our cell we have endoplasmic reticulum, but in case of muscle there are sarcoplasmic reticulum, because there is sarcoplasm. So sarcoplasmic reticulum inside your skeletal muscles are present abundantly. Lots of sarcoplasmic reticulum. Here they are also present, and here also present, but very less, right? Then what about the gap junctions? Gap junctions are those junctions, those gates, those molecular gates in between the adjacent cells, like epithelial cells have the gap junctions in between them, right? So in case of skeletal muscles, gap junctions are absent. In case of cardiac muscles, they are present, but they are present in the intercalated discs, which I will show right now. You can see on the screen, right? So inside the cardiac muscles, gap junctions are present. Inside the skeletal muscle, gap junctions are absent, and inside the smooth muscles, they are present in unitary muscle. Unitary means every muscle, every two muscle, right? In between every two muscle. What about the autorhythmicity? Autorhythmicity means that these muscles have the property of automaticity. Automaticity means that there are, for example, like cardiac muscles in our body have the tendency to auto-rhythmate itself, to auto-generate current, to auto-generate action potential, to automatically open sodium channels, right? So in our skeletal muscles, there is no auto-rhythmicity, right? The property of auto-rhythmicity in our skeletal muscles is not present, right? It requires a very important stimulation, which I will discuss later, which it requires the motor stimulation from the nervous system. That is must for it. But in case of cardiac muscles and smooth muscles, there is the property of autorhythmicity. They can generate lots of current, which will increase their contraction power. They can generate less power, right, which can decrease their contraction. So this property is present only in smooth muscles and cardiac muscles, and it is not present in the skeletal muscles. The next important property is about the thin filaments. What about the thin filament attachment? In the muscle, around the muscle or inside the muscle, there are thick filaments and thin filaments present, right? The thin filament of the skeletal muscles are attached to the Z line or Z disc. While I was explaining to you guys the sarcomere, the structure of sarcomere, there were two Z lines, inside and between the M line, in the thick filament, and above and below there were thin filaments. So those thin filaments were attached to the Z lines, right? So in case of cardiac muscles, skeletal muscles, the thin filaments are attached to the Z line or Z disc. In case of cardiac muscles, there are also sarcomeres present, and the thin filament is also again attached to what? Z line. But in case of smooth muscles, there are no sarcomeres, so those contain their thin filaments attached to special bodies which are called dense bodies, right? Next, what about the regulatory proteins? Let me go a little bit up. The regulatory proteins, right, which will regulate the contraction and relaxation of the muscle. In case of skeletal muscles, they are the tropomyosin and troponin. In case of cardiac muscles, they are also tropomyosin and troponin. But in case of smooth muscles, they are calmodulin, myosin, and light chain kinase. So those three are the proteins which regulate the contraction and relaxation of the smooth muscles. But tropomyosin and troponin are the regulatory proteins which regulate the contraction and relaxation of the smooth cardiac muscle and skeletal muscles. Next, what about the calcium source? From where the calcium comes inside the muscle cell, right? In our skeletal muscles, we have lots of storage of calcium which are present in the sarcoplasmic reticulum, right? In case of skeletal muscles, the calcium comes from the sarcoplasmic reticulum which is already stored there, right? But in case of cardiac muscles, they come from sarcoplasmic reticulum as well as from extracellular fluids. And in case of smooth muscles, they are always coming from the extracellular fluids, right? What about the calcium receptor? When calcium comes inside the cell, to which protein does it bind? In case of skeletal muscle, they bind to troponin of the thin filament, right? And in case of cardiac muscles, they will also bind to troponin of the thin filament, but in case of smooth muscles, it will bind to calmodulin of the thick filament. This is the difference. Next property, what about innervation? When nervous fires come, right, nervous stimulation comes, all our skeletal muscles are voluntary, right? So they are stimulated by somatic motor fiber. Somatic means voluntary, right? So they are innervated by somatic motor fibers. In case of cardiac muscles and smooth muscles, they have the autorhythmicity. They have the property of automaticity. They are innervated by autonomic nervous system fibers. Smooth muscles are also innervated by autonomic nervous system or autonomic nervous fibers, right? What about control of the muscle? Our skeletal muscles are voluntary. Our cardiac and smooth muscles are involuntary in nature, right? Can you stop the heartbeat of yourself? No. Can you stop the contraction of the cardiac muscles? No. Can you stop the contraction of those smooth muscles which are present in your blood vessels, walls? Can you stop the contraction and relaxation of those smooth muscles which are present in the GIT tubes? Can you stop the contraction and relaxation of all those smooth muscles which are present in your respiratory tube? No. So they are involuntary. Because they are involuntary, because they are supplied by autonomic nervous fibers, right? Autonomic fibers, so they are involuntary in nature. But in case of your skeletal muscle, look, if I want to raise my hand like this, right? So I have to use my bicep muscle. I have made the decision in my brain that I am going to raise my hand. So when I made the decision, from there motor stimulation came, motor fibers, motor innervation came to the bicep muscle. Bicep muscle is ready for contraction because motor fiber has reached here. Motor fire has reached here. Bicep muscle is ready for contraction, but I'm not doing it. I'm not doing any contraction. So if I'm not doing any contraction, muscle is ready, but I'm not doing contraction. So this is voluntary in my control. Right? So skeletal muscles are voluntary. Nervous stimulation required. Does your cardiac muscle require nervous stimulation? No. Does your smooth muscle require nervous stimulation? No. Why? I will tell you. Does your skeletal muscles require stimulation? Yes. Why? Because they are innervated by motor fibers. and because they are more innervated by autonomic fibers. Now listen carefully, one thing else, if you cut off these autonomic fibers, still your heart will beat, still your smooth muscles will contract and relax. Why? Because they have those sodium channels which can automatically open and will allow sodium to enter inside the cell and will allow the cell to take it to the threshold potential, and cell will start contraction and relaxation, so action potential will be carried. But in case of skeletal muscles, there are no such channels, not such sodium channels present, right, in your skeletal muscles. It requires all those channels to be stimulated by somatic motor fibers. Right, friends. What about next important points? What will happen to if you stimulate the muscle? If you stimulate the skeletal muscle, it will get excited. If you stimulate cardiac muscle, it will either be excited, either it will be inhibited. And in case of smooth muscles also, it will either be excited, either inhibited. Right. Mode of tissue repair. It means if you damage the muscle, what will happen to it? Either it will repair or not. So in case of skeletal muscles, there is less power of regeneration. Then what will happen if you damage a muscle, if you cut a muscle, what will happen? It will not easily regenerate. Fibrosis will occur. And in case of cardiac muscles, also fibrosis will occur. They have less power of regeneration. But in case of smooth muscles, they have good capacity of regeneration. Right? If they are damaged, those damaged cells are replaced by new cells through mitosis. They will fastly mitose, increase their number and regenerate again. Right. The next thing, what about the neurotransmitter used by the muscles for their stimulation or inhibition? All the skeletal muscles use acetylcholine. All the cardiac muscles use epinephrine and norepinephrine. All the smooth muscles will use all of them, acetylcholine, norepinephrine and epinephrine. So they use all of the three neurotransmitters. What about the response to stimulus? What does it mean? If you stimulate the muscle, what will happen? Response of the skeletal muscles are graded. Graded means the potential of the skeletal muscles will gradually increase. Right? But in case of heart muscles, if you stimulate one cell, that stimulation will cause all the cells to stimulate. That's why I have written here all or none. All or none means if you stimulate one cell, that cell through the gap junctions will stimulate all the cells. So suddenly all the muscle will contract and all the muscle will relax. So it means if you stimulate one part of the heart muscle, that will cause the stimulation of all parts of the heart muscle, and all the heart muscles will contract and relax. Right. In case of smooth muscles, they change their tone. What does it mean by change their tone? If you increase calcium influx, they will increase contraction. If you will decrease calcium influx, they will decrease contraction. Right? Next. The next property after response to stimulus, here graded, all, none or change tone, that is titanizable. What is meant by titanous? Titanous or titanizable? Look carefully. If you stimulate one muscle, right, many times, for example, you stimulate the bicep muscle with more and more motor impulses, so that lots of motor impulses will cause the muscle to either contract strongly or either relax strongly, right? So that is, this property is called titanizable. So our skeletal muscles are titanizable, right? Our cardiac muscles are not titanizable, and our smooth muscles are also more titanizable. Right? What does it mean by titanizable? You know, tetanus disease. In the tetanus disease, there is lots of motor impulses. Due to that motor impulses, the muscle will show the long period of contraction, so muscle will remain like this. Due to the frequent stimulation, the muscle will remain like this. Right? It will not relax. So this is called tetanus. Right? So skeletal muscles are titanizable and smooth muscles are titanizable. Cardiac muscles are not titanizable. Next, what is the work range? Work range means of work range of the skeletal muscles are at the peak of the length, work range of the cardiac muscles are in rising length, and work range of the smooth muscles are in length of the force curve. Right. Next, what is the postsynaptic receptor? Postsynaptic receptor means when the nerve releases the neurotransmitter, and this is a muscle, on the surface of this muscle cell, which receptor is present? So in case of skeletal muscles, nicotinic receptors are present. In case of cardiac muscles, adrenergic and muscarinic receptors are present. Right? And in case of smooth muscles, adrenergic and muscarinic receptors are present. This is the difference between the postsynaptic receptors. For example, this is the nerve and this is the muscle, and this is the synaptic cleft. Right? This is presynaptic membrane, this is postsynaptic membrane. So on this postsynaptic membrane, which receptor is there for this neurotransmitter which is released by this nerve? So here in the skeletal muscles, nicotinic receptor, in the cardiac receptor muscles, adrenergic and muscarinic, and in the smooth muscles, adrenergic and muscarinic receptors are present. Next, development of smooth sarcoplasmic reticulum. In the skeletal muscles, you have lots of sarcoplasmic reticulum. Here they have moderate, and here they have very little sarcoplasmic reticulum, because they don't need sarcoplasmic reticulum, because calcium will influx from the extracellular fluids, but they store calcium in the sarcoplasmic reticulum. What about the resting membrane potential of the skeletal muscles and cardiac muscles and smooth muscles? The resting membrane potential of skeletal muscles is minus 90 millivolt. And that of the cardiac muscle is also minus 90 millivolt. And that of the smooth muscles is minus 65 to minus 35 millivolt is the resting membrane potential. The next properties, so these were some properties that we discussed, the comparison between the skeletal muscles, the cardiac muscles and the smooth muscles we discussed. So these were some properties, friends, that I shared with you. I hope you enjoyed the lecture and I hope you liked the lecture. Now in the next lecture we will start discussion about the muscles of the upper limb. Each muscle I will discuss about its location, about its actions, about its blood supply, venous drainage, lymphatic drainage and innervation. So see you guys in the next lecture. Till then, Allah Hafiz.